Good morning, students. Welcome to the biology class. We are going to continue. In the immunity part of this chapter, we discussed about the various types of immunities, components of immunities, and today we are going to discuss about the antibodies. What are antibodies? They are the glycoproteins called immunoglobulins. There are 5 classes of immunoglobulins, and each antibody or immunoglobulin is specific in their function. For example, IgA. It is present in all body secretions including mother's milk and colostrum. Colostrum is the first milk of the mother's milk, and that is very important and very effective to the infant. That's why the breastfeeding is being recommended. Next is IgD. It is effective against the toxins, means any harmful substances if they enter, and allergens, that is again the foreign particles. IgG, it can pass through the placenta. IgM is first to reach the site of infection, and IgE, it is the mediator in the allergic response. So the antibodies are the glycoproteins and they play a very specific role. Structure of antibodies. Each antibody has 4 polypeptide chains held together in the form of Y-shaped structure. 2 of polypeptide chains are long and heavy, called as H chain, while the other 2 are short and light and called as L chain. Hence, an antibody is represented by the formula H2L2. The tips of 2 upper arms are bound to the antigen in lock and key manner to form the antigen-antibody complex. Attachment and the bending occur by the means of disulfide bond. An antibody has variable portion in the arm called V region or antigen binding fragments. The remainder of antibody is called the constant portion. So in this picture, that structure will be more clear to you. These are the 4 polypeptide chain, this and this one. These are your 2 heavy chains, and this one and that one, this is your 2 light chain. So H2 and L2, and that gives the Y-shaped formed. So the 4 polypeptide chain, 2 are heavy and 2 are light, and they are giving you the Y-shaped structure. And this one, this is the antigen specific site where the specific antigen will bind with the antibody and form the antigen binding site, which is known as V, means variable, and C is constant, that is both in the heavy and the light chain. And this is called as a bilateral activity site, and they are held together by the disulfide bond. So this is the structure of your antibody, which is represented as a Y-shaped structure, and this is very important as the examination point of view. Next, and the very important topic is your vaccination and immunization. Edward Jenner coined the term vaccine, and vaccine means cow. What is vaccination? Vaccination और immunization दोनों में क्या डिफरेंस है, that is very important. Vaccination का मतलब है कि जब हम बॉडी के अंदर inactivated pathogen डाल देते हैं, और the body means the person is going to give the protection against the disease. This is the vaccination. Vaccination का मतलब है कि introduction of weakened or inactive pathogen or proteins into a person to provide protection against the disease. And what is immunization? Immunization is a process by which the body produces antibodies against the vaccine and develop the ability to neutralize pathogens during the actual infection. So what is the major difference? Vaccination का मतलब है कि पैथोजन inactive form में बॉडी के अंदर इंट्रोड्यूस करना. आफ्टर द इंट्रोडक्शन जब बॉडी उस पैथोजन के अगेंस्ट एंटीबॉडीज बनाती है और बॉडी को डिजीज फ्री करती है, दैट प्रोसेस इज नोन एज इम्यूनाइजेशन. कभी कभी अगर हमने बॉडी के अंदर या किसी पर्सन के अंदर वैक्सीन लगा दिया पर उसने एंटीबॉडीज नहीं बनाए तो उसमें इम्यूनाइजेशन नहीं हुआ, उसमें क्या हुआ? सिर्फ वैक्सीनेशन. वैक्सीनेशन का मतलब है बॉडी के अंदर वीकेंड पैथोजन डालना जिसके अगेंस्ट बॉडी एंटीबॉडीज बनाए और बॉडी अपनी बॉडी को उस पैथोजन के अगेंस्ट प्रोटेक्ट करके रखे. पर अगर सिर्फ वैक्सीन डाली गई, मतलब सिर्फ हमने पैथोजन डाला, तो दैट इज द वैक्सीनेशन. पर अगर बॉडी ने उस वैक्सीन के अगेंस्ट एंटीबॉडीज बना ली और न्यूट्रलाइज कर दिया उस पैथोजन का जब एक्चुअली उस इन्फेक्शन ने कॉज किया, तो उससे हम बोलेंगे इम्यूनाइजेशन, जो हमने प्राइमरी और सेकेंडरी रिस्पॉन्स में किया. प्राइमरी रिस्पॉन्स का मतलब वैक्सीनेशन डाल दिया और इम्यूनाइजेशन हो गया, बॉडी ने एंटीबॉडीज बना ली. और वही एंटीबॉडीज तब काम आएंगी जब एक्चुअली इन्फेक्शन बॉडी को होगा और बॉडी उस इन्फेक्शन से अपनी बॉडी को बचा लेगी और उसे बीमार नहीं होने देगी. दिस इज व्हाट कॉल्ड एज वैक्सीनेशन एंड immunization. So the principle of vaccination and immunization is based on the property called memory of the immune system, that is a unique feature of acquired immune system. The vaccine generates memory B and T cells that recognize the pathogen on the subsequent exposure and produce the intense immune response. कि उसने पहचानना है, recognize करना है, and then accordingly they are going to respond and make the body free from the infection. This is normal active immunization. If quick immune response is needed, as in tetanus infection or snake bite, performed antibody means ready-made antibodies or antitoxin is injected into the patient. This type of immunization is called passive immunization. As we said, that is the passive immunity. By recombinant technology, vaccines have been produced on the large scale. For example, hepatitis B vaccine is produced from the yeast, and nowadays we are also searching for the vaccine of coronavirus by the same recombinant technology. The next is lymphoid organs. Lymphoid organs are the organ where the origin and or maturation or proliferation of the lymphocyte takes place, means multiplication of lymphocyte takes place. It is that particular organ जहाँ से वो origin होंगे, जहाँ mature होंगे, जहाँ multiply होंगे, उन organs को हम क्या बोलते हैं? Lymphoid organs. And they are of two types, primary lymphoid organs and secondary lymphoid organ. प्राइमरी लिम्फॉइड ऑर्गन्स हम उन्हें कहेंगे जहां पे द ऑर्गन्स दे आर गोइंग टू बी ओरिजिन, दे आर गोइंग टू बी मैच्योर, पर जहां वो एक्शन करेंगे, जहां वो एंटीजन को खत्म करेंगे, दैट इज व्हाट कॉल्ड एज द सेकेंडरी लिम्फॉइड ऑर्गन. सो फर्स्ट वी विल डिस्कस अबाउट द प्राइमरी लिम्फॉइड ऑर्गन. प्राइमरी लिम्फॉइड ऑर्गन आर दोस where the immature lymphocytes undergo maturation, differentiation into the antigen specific lymphocyte. Means they will become mature, they are going to be developed, they are going to be differentiate, and the two important primary lymphoid organs are bone marrow and thymus. Bone marrow, it is a main organ where all types of blood cells, that is your RBCs, WBCs, plasma, including your lymphocytes, they are going to be mature. So bone marrow provides micro environment for the development and maturation of B lymphocyte. That is, they get matured in bone marrow, that's why the name is given as the B lymphocytes. So next is your thymus. it is located beneath the chest bone near the heart. So this is the location of the thymus, that is beneath the chest bone near the heart. This gland keeps reducing in the size with age. It provides microenvironment for development and maturation of T lymphocytes. That's why the name is T lymphocytes. So this gland keeps reducing in size with the age. That's why the factor of aging is, as the age increase, immunity starts decreasing, and because of the decrease in the immunity, that leads to the aging. So this is one of the factor of the aging. Next is your secondary lymphoid organs. Secondary lymphoid organs are those where the lymphocytes interact with the antigen and multiply to become effector cells. Effector means what effect they are going to give to that particular antigen. The example is spleen, lymph nodes, tonsil, appendix, and pair patches of your small intestine. So first we are taking spleen. Spleen contains lymphocytes and phagocytes which used to engulf the antigens. This is the position of the spleen. It acts as a filter of the blood by trapping the blood-borne microbes जो blood को infection करता है, उन antigens को उस blood को filter करना. It also a reservoir of the erythrocytes, and it is also being known as the graveyard of RBCs, because when the life of RBCs is being completed 120 days, the debris of the RBCs are there in the spleen. So it is also being known as the graveyard of the RBCs. The next is your lymph nodes. The lymph nodes, that are the small solid structures found at the different points along the lymphatic system, that is your lymph vessels, and they act as a filter and trap the microbes that have entered the lymph, which are giving infection to the body fluid. Antigen trapped in them activate the lymphocytes present in the lymph nodes and produce the immune response. The next and the last one is your mucosal associated lymphoid tissues, and lymphoid tissues located with the mucosal lining of major tract, that is your respiratory, urinogenital, digestive tract. They are called as the mucosal associated lymphoid tissues, and that particular microorganism get trapped in that particular mucosa and give the immune response. It accounts for about the 50% of lymphoid tissues in the human body. So mucosal associated lymphoid tissue is also being known as a MALT, M-A-L-T. So this is about your lymphoid organs, that is your primary lymphoid organs, secondary lymphoid organs. So today we discuss about vaccination, immunization, and the structure of antibodies. Next lesson will be on the immune disorders, in which we discuss about the allergy, about the autoimmunity, and the AIDS. So till then, goodbye, stay safe, stay home, stay healthy. Thank you.